Antra, we can start.
Good evening, everyone. I'm Antara Ghosh, and I'm currently pursuing my bachelor's degree in forensic science from Parul University. We are delighted to have you all join us for our webinar series, Future Tech in Forensics, a dive into past. This webinar series is presented in collaboration with the Department of Forensic Science, Applied Sciences, Parul University. This webinar series focuses on the recent development and the advancements in the forensic science field. Today is our seventh webinar. Forensic drug analysis involves the identification and quantification of substances in biological samples for legal purposes, utilizing various analytical techniques such as chromatography and mass spectrometry. Analysts determine the presence of drugs, their purity, and concentration. This discipline aids law enforcement in criminal investigations, drug related prosecution, and ensures the accuracy of toxicology reports in cases involving substances, abuse, or poisoning. All the participants will be provided with a link to the feedback form in the chat box of the webinar session. On filling the form, they will be given an e-certificate for the participation. If any participant has any queries related to the topic, feel free to ask in the chat box or in the YouTube comment section. The topic of today's session is advancements in forensic drug analysis and opportunities of rapid screening from powder to pill by Sheetal Rajan. Ma'am. To invite and introduce the expert, I would like to extend an invitation to Mr. Arun Kumar Kavat, CEO and founder of Forensic Talents India. Sir.
Thank you, Antra, for your introduction. Our today's speaker is Sheetal Rajan. So, basically, Ms. Sheetal Rajan is distinguished expert in the field of forensic science with a comprehensive academic background and passion for research. Armed with honors degree in BSc in Forensic Science from Jain School of Science, Jain University, Bangalore. She has also completed her master degree in Forensic Science from the specialization with Forensic Chemistry and Toxicology from National Forensic Science University, known as NFSU. She has previously, just a second. She has previously worked in the sector of forensic drug doping, date rape cases, as well as in the field of drug poisoning in esteemed institute and in forensic science laboratory, Ahmedabad, Gujarat. Essentially, she has displayed a deep interest in academic writing and has published paper and chapters in the forensic physics as well as in general forensic sciences. She is currently working as assistant professor in Parul University, Gujarat, India. With keen interest in pursuing the boundaries of knowledge, she dedicated to concluding groundbreaking research that advance in field of forensic science, unraveling mysteries and shedding light on complex criminal cases. She would like to grow as an academician in forensic science to help students and engage in the field of forensic science with new research in futures. We are very happy to have you, ma'am, in our platform. Ma'am, you can
Thank
share your
you.
screen.
Yes, sir. Thank you for introducing me, sir, and thank you, Antra. I'll be sharing my screen now. Please confirm if you're able to view it.
Sure.
Sir, if you could stop sharing your screen, I would be able to share
Sure,
mine as well.
sure.
Is the screen visible?
Yes, ma'am.
Okay. Once again, good evening to one and all present here. My name is Sheetal Rajan. I'm currently serving as an assistant professor of forensic science from Parle Institute of Applied Sciences, Parle University. So, the topic for my webinar is Advancement in Forensic Drug Analysis, where I'll be specifically localizing my research for opportunities of rapid screening from powder to pill. Are you able to see the next slide?
No, ma'am. It's on first slide only.
And now?
Yes, ma'am.
Yes, ma'am.
So, before we start the topic, I thought we should essentially start with a very good quote. And so, by Thomas Huxley, there's a very famous quote. It says that the man of science has learned to believe in justification, not by faith, but by verification. And this isn't essentially, which is something that's only applied in forensic science. As far as scientific analysis is concerned, whether it's in the field of forensic toxicology, whether it's general pharmacy, whether it's to find out a solution to any problem, for that matter, you do use a scientific approach. So you don't just believe in something that you see and say that this is so and so. You believe it if it's justified with some scientific background. So I thought I would start with a quote. So we're talking essentially about drugs, and now when the word drug comes to our head, I'm very sure a lot of students, especially um, as per my experience, they've told me, "Ma'am, that's a very bad thing. That's something that we shouldn't consume." And I would say not likely. A drug can be good, a drug can be bad, right? So, in general, if you define a drug, it is any substance, okay, or it's a product of a source, which is actually intending to modify a certain function of your body. So, your body functions in a certain sequence. Either a drug may disrupt the system, either it might enhance the system, it might act as an antagonist, that is, reversing the effects of the system, and so on. But here it is producing some change to the system, right? So any substance that when ingested through any means, when it brings about a physiological effect into your system, that substance is called a drug. And this was wonderfully explained by WHO. But where did the term drug come from? That's from a French word which means drogue, which is actually a dry herb. So now, again, explaining why certain drugs could be good for you and certain drugs could be bad for you, we have very, like, I would say, prominent words over here. One is prescription drugs, okay, and the other one is non prescription drugs. So, in other words, you call them as OTC. Now, prescription drugs, just as the name suggests, it means any drug that you purchase from a pharmacy with a prescription. That is a written statement from a doctor. If you have any ailment and the doctor tells you you need to consume so and so within 2 days, within 3 days, 2 tablets per day, that's a prescription. That's signed by a doctor. A non prescription drug is also known as OTC, which is over the counter, which most of us have experienced in, I would say, in terms of purchasing. When you go to a pharmacy and you say that I have a headache and I would like to have some, pain relieving tablets, essentially the pharmacist would give you a packet of Dolo. And Dolo is nothing but paracetamol, which is actually used to relieve pain. So for that, did you have to give any prescription? No, it's over the counter. No questions asked, but answers are given, right? So, coming to a specific class of drugs, I clearly mentioned here something known as therapeutic class of drugs. So, good drugs are therapeutic, bad drugs are, well, bad. So, therapeutic drugs can actually turn into bad drugs if it's ingested beyond a certain dose, that is, if it's abused. So, we have analgesics here. Analgesics are nothing but drugs which help in pain relief, like the example of Dolo that I've given. You have antibiotics. Antibiotics are drugs which are against any bacterial infection. So essentially, a person who has had food poisoning is more likely to have an antibiotic. You have antihistamines, that's for people who have allergic symptoms. It basically acts against the effect of histamine, which is producing symptoms of allergy like sneezing, coughing, and dryness of throat, etc. Then you have other drugs which may fall into different classes like cardiovascular drugs. You have anti-diabetic drugs, which unfortunately it's consumed a lot today, and so on. So, what are the sources of drugs? Where do these drugs come from? Are they natural? Are they chemically modified? Is it something that you probably find in, I would say, good access? Is it what is the source for the likelihood of an active substance to be accessible to people? So that comes into the next slide. That is your different sources of drugs. So I've mentioned a few over here. You have your mineral sources like liquid paraffin. Now, liquid paraffin is actually refined mineral oil. It's mostly used in most medicines, I would say. Then you have kaolin, which is an aluminosilicate powder. It essentially looks like a clay, a very hard-bound clay, and it's used, I would say, in medicines, in cosmetics, as well as in toothpastes, right? But this is also a source from which you can retrieve drugs. Then you have magnesium sulfate. These are actually dry, colorless crystals, which can be used as an anticonvulsant. So, convulsants are those drugs which treat seizures. So, if there is a patient who's suffering from convulsions or any sort of erratic body movement, essentially you give a very safe drug that is magnesium sulfate. Then you have sources of animals, which is, I would say, probably a bit surprising to most of us. So you have insulin. Now, insulin is actually retrieved from the pancreatic system, the pancreas of cows and pigs, etc. These are the organs from which you would retrieve insulin from. And anti-toxic sera. Now, this is also retrieved from the intestine of pork. And then you have heparin. Now, heparin is, I would say, kind of a savior in forensic science, and I would say in most cases of blood preservation, it's used as an anticoagulant. And heparin essentially would, I wouldn't say deplete it, but it would completely prevent the blood from getting thicker in state. A very valuable thing about antitoxic sera is that antitoxic sera would contain most amount of antibodies which would fight against certain infections. These can be retrieved from either, in some cases, mostly from humans as well. But as far as ethical issues are concerned, we don't go for antitoxic sera from human beings. Then you have plant sources. Plant sources like morphine, atropine, etc. Morphine is a product of opium. It's an opiate, so it's actually used for pain relief as well. That is intense pain, especially during surgeries. And atropine is actually an anti-motility drug. It is used for relieving the symptoms of diarrhea. Then you have sources like microorganisms, where you have penicillin as a great example, which essentially would ward off any sort of infection. You also have synthetic drugs, just as I've explained, analgesic drugs, and then you have hypnotic drugs. And hypnotic drugs, I would say, they induce a sense of sleepiness, a state of hypnosis. Um, I wouldn't say complete hypnosis, but I would say that it brings about effects of sleepiness, drowsiness, and so on. Now, if you are an individual who wants to know about different sorts of drugs which are present, what are its effects, what are its formula, even formula with respect to understanding the chemical constituents of a particular drug, you can go for a book. I would say a legalized book known as pharmacopoeia. So different countries have established different pharmacopoeias, which talk about the different types of drugs which may be there under each alphabet, under each chemical constituent, under each category of its effects, and so on. So I would suggest any of you, whoever's interested in looking into what do drugs contain, or what does a specific herbal drug contain, or specific synthetic drug contain, you can definitely go for something known as a pharmacopoeia. And I'm pretty sure if you're in the university level, most universities are equipped with a pharmacopoeia. So I would suggest to give it a look. Now, this is something that I would say on the negative side. You talk about effects of drugs in a forensic sense, then definitely it is going to be negative. So, if I have to vaguely describe forensics, just as Andra had explained, you are trying to, find an answer to a specific cause, that is a specific cause of whatever criminal activity has taken place. So, with respect to any crime where drugs are involved, essentially, if the victim has been subjected to a certain exposure of drugs, either through veins, either orally, and so on, it so happens that the person does succumb to it, and eventually, when the person is, that is, the person's loved ones or anybody at the scene of crime, is essentially calling the law enforcement agencies to come and investigate, you are more likely to see a negative effect of drugs. So, I've written one sentence here, and that pretty much sums up everything. That is, your drugs, they affect the way you see, smell, think, hear, move, and sleep. So, in this case, if I have to say the way you see, there are drugs which completely differentiate a person from reality and fantasy. People begin to see colors, like, and hear colors even. And that's a very odd statement, don't you think? Hearing a color, but that's what happens when people do consume, I would say, wave drugs and club drugs like LSD. So in that case, we are not able to differentiate between what is fantasy and what is reality. In this case, what they're mostly going to do is they will be, in the zone of under the effects of drugs. So they begin to hear colors, they begin to see the shapes of people around them changing, and so on. The way they smell changes, the way they think definitely changes. And I think this is a very important fact to establish. The use of drugs can change the way a person thinks at that moment of time. And unfortunately, there are so many drugs which also wipe out what they've thought right after the event has taken place. Like, I would say a very good example would be any date rape drug, which is used to, completely take out the memory of an individual. And there are such cases which exist where drugs like ketamine, GHB, etc., have post risk of amnesia. So after the effect of the drug has worn off, the victim is no longer able to retain what has taken place. So that is a scary fact, and that actually happens. And the way you move, yes, definitely it affects motility and the way you sleep as well. So here, I would say there is both a good and a bad aspect here. With respect to sleep, there are drugs which are prescribed by doctors to improve the quality of sleep. But if such drugs are abused, or if such drugs are taken more in quantity, or if they're taken frequently, more than what is prescribed, it is, I would say, it is going to probably affect them at a fatal stage. So that's what effects of drugs are concerned with. Now, as a forensic scientist, you are most likely going to come across cases where drugs are involved, right? So I've taken two case studies. The very famous case study is that of Michael Jackson. So Michael Jackson was a very prominent, I would say, pop figure, but the cause of death of Michael Jackson was supposedly overdose of a specific drug, that is a drug known as propofol. And propofol was actually a drug which was administered, or which was prescribed to Michael Jackson by his own doctor, because Michael had actually faced issues with sleep, and on prescribing basic drugs of benzos like like I've mentioned over here, lorazepam, diazepam, injections of benzos as well, like midazolam. These did not exactly work. And let me be clear with you, any drug which is intravenously pumped into your system is most likely to get absorbed much faster, and the effects are also going to be really, really intense in comparison to someone who has eaten the drug, that is, through oral consumption. Because what happens in this case is, a person who essentially takes the tablet is going to undergo the different process like absorption, distribution, metabolism, distribution, etc. So in that case, what happens is it's going through several layers, going through several, sheets. So in that case, it's either, if the person is lucky, it could be that not the entire drug gets absorbed into the system. But in the case of intravenous, that is through your veins, if it's injected, it will produce effects much faster, and it is more likely to pose effects that might affect a person's health poorly. So I would say a very good example where intravenous method is very good is in the case of emergency, when the person needs an intravenous method of waking up from a state of unconsciousness, or in that case, what you do is, the person cannot obviously only take a tablet, so you will intravenously inject the medicine into their system. So this is one of the prominent cases that we've seen in history, that is the use of a drug actually posing a harmful risk to the patient. And in this case, the doctor was actually taken into custody. So I would say the penal code is pretty strict with its regulations. The next case that we're looking into is, I would say, a very common case. It's something that I think all airport officials must be going witnessing every day. That is drug trafficking cases. So there are groups of people who think they are extremely intelligent in smuggling goods, and they do find creative measures in order to do so. So um, I'm sure that you guys must have heard about the Spanish drug war, which happened years ago during the time of Escobar. So at that time, apart from smuggling drugs through, vehicles and through people, they used to do it by air. And how they would do that is through creative ways. They would essentially make the person swallow those pills and then smuggle them, so that it doesn't get detected by any system, or in shoes, taped to the clothing, and etc. And there are some people who don't hide the drugs as much. That is, they keep it in suitcases, but they think that certain detectors or certain systems wouldn't be able to detect packages because of the layer of clothing on top, or maybe they keep it in normal food containers as if to show that this is simply food, or if it's a cosmetic, and so on. So what happened was MDMA, that's a very strong, I would say, club drug. MDMA pills, or MDMA powder, which is usually pulverized pills, was in packages. So these packages were essentially caught by the law enforcement agencies at the airport. And now this is something that I would be explaining at a later stage. But what happens is, generally in a case of crime where you see powder, um, it's just like how you would approach a scene of crime with blood. When you go and see a red liquid, you will first ask yourself whether that's blood or not. The same thing applies to a forensic chemical crime scene. I would say if you see white powder, people shouldn't be quick to assume that that could be drugs, but people shouldn't be quick to assume that that cannot be drugs. So you need to play a role there where you would go with caution, and you would collect every evidence that is relevant to the crime. Now, another quote that is something that I've already mentioned in the beginning. There is obviously an extremely thin line between medicine and poison. So, everything is good if you follow a safe dose, if you follow a safe frequency of dosage, if you follow, I would say, not taking the medicine every time you have that ailment. That is completely safe. But the dose makes the poison. That is, if a person has abused a drug, abusing a drug means nothing but taking it frequently more than what's required. If they are going by a longer duration of time when they shouldn't be doing so, that's when a drug can turn into a poison. So, simple substance like Dolo, for example, something that can be taken twice a day without any prescription, can pose a fatal effect if the person has taken it continuously. And so, the poisoning turns a little mild, and then with, I would say, a longer stage of with a longer period of time, it tends to become chronic. So, how does drug analysis actually take place? Now, as far as what I'm experienced in, as far as laboratory is concerned, most of the work of analysis of drugs is done in the lab, especially with respect to drugs. Because when you look at the general appearance of drugs, unless and until it gives out a sufficient, I would say, amount of odor, if it's distinguishable by color, because there are several drugs which are, white pills, but not every white pill might belong to the same class of drugs. So you cannot just see a pill and automatically decide that that could be so and so drug. So most of the work in terms of analysis is done in laboratories. But how your analysis takes place, the quality of your analysis, the precision, the accuracy of analysis will depend on how the forensic experts have collected, packaged, and preserved the crime scene. It's extremely important that when you step into a crime scene, you do follow ethical practices, you wear the right um, personal protective equipment, you will deal with the crime scene as if it's the only thing that's in front of you. And that's how it should be done. You will completely focus on the objects around you, see what is its linkage, if there is any connection between the bottle and the pill. That is also extremely important. So, drug analysis will fall under two specific branches that will fall under chemistry as well as toxicology. So, I would say, in simpler terms, if you want to understand what the drug is, what is its chemical composition, and to some extent, I would say what are its effects, etc., you will deal with that in the case of chemistry. If it's a narcotic drug, if it's psychotropic substance, these are all necessarily dealt with under chemistry. But if there is a case of poisoning because of drug, you will essentially go for toxicology, because toxicology does deal with the effects of poison, does deal with the composition of poison. Basically, it deals with any substance which can act as a poison. And as you know, every substance can act as a poison. It highly depends upon the dose. Even water, for that matter, can act as a poison. So there is a collaboration between the Department of Toxicology or as well as the wing of chemistry. They go hand in hand. And I would say the crime cases with respect to drugs being involved, it's huge. You have several crime scenes like homicides, where a person would administer a drug into somebody's food and then try to poison them, right? Like, for example, I would say cyanide has been unfortunately mixed with chocolates and been given to children in order to kill them quickly. And in the case of cyanide, the thing is, the effects of cyanide are quite intense, it is quick, and the effects of poisoning that is seen on the system is on the outside a bit lesser. So people wouldn't suspect that, oh, this child has been poisoned. It's a later effect. So that can be the case. It could be accidental poisoning. In this, again, I'm going to bring the example of children. Um, Kids don't know how medicines work, and they might think that, oh, medicines are pills. Pills look like candy. Now, if you've seen every medicine capsule, cabinet, or bottle, You must have seen keep this out of reach from children, which is to avoid any case of accidental poisoning or accidental drug intake by children. Most of the case of accidental drug intake has been done by kids statistically because of parental neglect. So that is one case to look at. Then you have suicidal as well, where most of the cases, just as I've mentioned, where the person takes the drug and succumbs to the effects of the drug in order to kill themselves for reasons that might be personal to themselves. And essentially, a lot of patients who have suffered from depression have known to commit suicide by the use of the same drug that was used to treat them in the first place. So, a person who has abused antidepressants has actually passed away. So, when you're collecting drugs in general, drugs may be present in different forms. You can find syringes in most cases, you can find spoons as well in most cases, where the person essentially melts the drug and then tries to snort it or tries to orally take it as well. You will find pills, you will find powders, etc. So the collection procedures for each is very important. You will use a good packaging technique as well to avoid any spillage of the contents. And overall, you're trying to maintain the chain of custody in this scene. And preservation as well, to protect the integrity of the evidence, you need to preserve it. And finally, when this entire evidence is forwarded to the lab, you will conduct analysis. Now, this is just a chart explaining drug analysis. So, what you do is you will get the drugs from the crime scene, you will extract the drug, you will extract the active compound. That is the effect of the drug that is pronounced is usually coming from the active component of the drug. Because not if you see drugs composition in general, it's not only going to contain the active components, it can contain dyes, it can contain a smaller quantity of impurities, then it can contain binders and so on. So you are essentially just looking at the active component, right? So extraction procedures do take place. You go for solid phase, liquid extraction, and so on. Because sometimes, even in the case of, I would say, food which is mixed with drug, you need to essentially take out the uh, drug particles or the drug molecules from the food matrix, right? So you will essentially go for extraction techniques and you'll perform an analytical technique. Now, analytical technique can give two results. It can either give you a yes or a no based result. That is, yes, it is present, or no, it is not present. And it can also give you how much is present in that specific sample. Or if the analytical technique is not supporting that case, it could also be inconclusive. That is, it is not giving you a proper result. So, most of your drug analysis follows the area of biological testing. Okay. Then you have chemical testing methods and you have instrumental testing methods. So, essentially, in the forensic sense, I would say all of these are equally important. There isn't anything like, oh, the biological analysis will overpower chemical or vice versa. In fact, all of us do aim at getting the answer to what drug is contained is present in a specific sample, and that's about it, right? At the end of the day, we aim at receiving justice. So as far as biological drug analysis is concerned, most of the traditional tests also are equipped with certain techniques where they use samples like urine, blood, hair, etc. So UDT, otherwise known as urine drug testing, it's usually used to test for drugs which might be present in urine. So, in this case, if the person has consumed the drug, it usually gets metabolized as well. So, metabolism is essentially breaking down the drug into simpler constituents. If the drug gets metabolized, then the effects of the drug are going to be less severe because it gets out of the body. So, it's not going to stay in the body for too long, and hence that's a good source of testing as well. If a person has if it's known that the person has ingested a specific drug, you will essentially go for a urine drug testing method where you will collect the urine samples of that person and you will be using certain indicators which might detect either a positive or negative of the drug. So there are usually strips, there are usually agents as well, or there are usually kits which come um, scene today. That is certain kits for specific drugs. So I've mentioned over here benzodiazepines, amphetamines, cocaine, marijuana, etc. The drug kits for these are different. They are actually modern methods of analysis. That's the advancement which we're talking about. So in this case, what happens is it's a kit which has a control column as well as a testing column. So in this case, if you put the urine sample by means of a dropper, if it gives you two lines, it's most likely going to be a positive state. But something to take care of is some color tests or some of these kits are good enough to confirm results if it's a rapid screening, but I would suggest, and as far as what I've studied as well, it is important to confirm these results using an instrumental technique. So this is going to be giving you a quick result. So this is at least giving you a sort of capability to detect something in the first place. So it's a great technique, I would say. Then you have blood drug testing. This is something extremely common in the clinical scene. In the clinical scene, again, the blood is taken out from the person and is essentially subjected to certain kind of tests to detect whether the specific drug is present or not. Hair follicle drug testing, which is something which some people might be aware of, some people might not be aware of. In this case, what happens is the hair follicles essentially would contain traces or elements of the specific drug which is consumed. Even nicotine to an extent can also be present in hair, which is what can pose as a challenge in that case. In that case, what happens is if you have to differentiate between an active smoker and a passive smoker, there is a passive smoker is a smoker who doesn't really smoke, but they're exposed to the smoke which is produced by active smokers. You wouldn't be able to essentially differentiate it that much unless and until you go for a very specific quantitative technique. So in this cases, you can test for amphetamines, opioids, and different classes of drugs as well. So specific chemical tests which you can do in the case, I've put one example over here. Heroin, morphine, codeine, these are all alkaloids of opium. Opium is actually known as, it's actually from opium poppy. So the drugs which you usually retrieve from opium poppy, they are known as opiates, and they don't usually go for any chemical modifications. The drug as is taken and is tested. So in this case, you have certain color tests like Marquis reagent and Mecky's reagent, frauds as well. In this case, if you see heroin, morphine, codeine, etc., they give about the same color. Now, as a scientist, how exactly are you going to say that, okay, this specific alkaloid is basically morphine, or this is probably papaverine, or this is probably heroin? You will not be able to say it as much, especially if the color change is the same. Similarly, in the case of Mekis, if you see over here, heroin and morphine, as well as 6-acetyl morphine and acetylcholine, they're giving you the same colors. Again, are you able to exclusively distinguish between each alkaloid? No, but that's where an instrumental technique comes in. So there are certain, I would say, challenges when it comes to color tests. A color test is just a presumptive test. It is not going to exactly confirm your result. It's going to give you a presumptive kind of screening capability. That is, it's just going to tell you yes or no, this is present. But in order to confirm that, you need a instrumental technique, because this is most likely going to give you either a false positive, that is, it's going to say positive even though it's not present in the sample, or false negative, that is, it's present in the sample but it is not in sufficient quantities to be detected, or if it's suppressed by other ingredients. Now, going to instrumental drug analysis, that is what I would like to take care of today. So you have several instruments or certain automatic machines which will essentially play the role of analysis of drugs. So what we have used for a very long time, and we still continue to use, because as far as I'm aware, and I'm sure most experts are aware of, instrumental techniques are always a boon. So they are essentially always going to give you results that are satisfactory. So if in fact I would say no manual labor is required, the drug is simply injected into the system, or it's subjected to the system, and then you get a reading on the basis of certain um, I would say, principles. So what we have used so far is UV visible spectroscopy. We've used GCMS, which is gas chromatography mass spectroscopy. Then liquid chromatography mass spectroscopy, which is LCMS. However, there have been certain advanced techniques like high resolution MS or high resolution mass spectroscopy and miniature ambient ionization MS. So let's take a look at each. So I have actually gone through several articles where several, I would say, scientists have actually taken different samples from different matrices using a specified condition of samples as well. I would say the ages of people were obviously restricted. The kind of samples that they required also had some sort of historical screening and so on to make sure that the person does not have any health condition. So these were some of the, I would say, precautions which were taken by the experts. But as far as analysis is concerned, we mostly always go for determining drugs or detecting drugs. And if you're detecting drugs, you're basically doing answering the what drug is present. That's the question which is answered. But when you're also going a little deeper into the topic, you're also specifying how much is present, right? Because just as I said, the dose makes the poison. That is, dose is quantity at the end of the day. So if you have to see that, suppose in the case of suicide that I've mentioned, a person has taken antidepressants. Now, antidepressants is actually their medicine, which they're supposed to take to cure depression. But there is a specified dose. If they go beyond that, we should also establish if somebody else has given it to them and then they have been hung, you know, in a case of homicide, or if it's self administered, or if they've actually tried to commit suicide or have successfully, unfortunately, attempted in committing suicide using the same medicine. So their quantity plays a great role. So here you have a study which is determination of drugs in hair, like I've told you, biological along with chemical sciences. Determination of drugs in hair using GCMS-MS. So here the study was done to detect several drugs like flunitrazepam, then tramadol, azaprone, these are benzodiazepines or antidepressants. So what happened here was there was some sort of sample treatment that was done. Sample treatment is nothing but preparing the samples for analysis. So basically, it's like in simple terms, I would say you're basically making the batter so that you can keep it in the oven and bake a cake. So preparing the batter part is the sample treatment part, where the drug is usually treated with N-hexane in this case. It is usually rinsed as well and is also cut so that it is suitable for analysis. So this was actually subjected to GCMS. Now, GCMS is an instrumental technique which depends on affinity of that substance towards either the stationary phase, which is going to be non-mobile, and the mobile phase, just as the name suggests, a solvent phase. So what gas chromatography does is it usually takes a sample and it will allow the sample to flow through by the use of gas. So gas is usually pushing the sample. So the mobile phase which is used here is gas, unlike your other techniques where the mobile phase would be liquid in nature. So here only volatile substances can be treated using GCMS-MS. So in this case, that could be kind of a disadvantage. So essentially, your antidepressants, in order to be made volatile, they need to be derivatized. And derivatized is usually a step which you do to treat the sample so that you can change the chemical composition so that you can make it more volatile. So basically, you're enhancing the properties of that specific drug so that it can be used for analysis using GC. That is one case. So this was also done. Then another technique was using LCMS-MS. In this case, the person had used, I would say, plasma or serum samples, and they have used actually active cells from the plasma as well. In this case, they had gone for solid phase extraction techniques because in case of solid phase extraction techniques, it was able to absorb more ionic traces of the compound. So that was essentially more superior than your traditional liquid-liquid extraction. Solid phase is nothing but using a solid sorbent, whereas liquid-liquid extraction is using two immiscible liquids. So it's just an extraction technique. So here, extraction technique played a great role in analysis using SPE. And this was actually considered to be a very rapid and robust technique as well, because in this case, multiple drugs could be analyzed. So this was also a very good technique, which was, I would say, used by these famous scientists. Then going into UV visible spectroscopy, it's one of the simplest techniques to use, where you would usually add the sample containing drugs into a cuvette by means of a cuvette, and you will screen that in a specific UV visible range, right? That is 200 to 400 nanometers, and from 400 to 800 nanometers. So in this case, the drugs are screened entirely throughout the entire UV visible range, and then they're checked for, its peak. So whichever drug absorbs at a specific wavelength at a nanometer range, it will produce a peak. So when you essentially plot a graph between wavelength and absorbance, you can see the peak or the lambda max. That's a maximum wavelength which is absorbed by the substance. And accordingly, you can also measure its concentration. So because it's directly proportional to its concentration, you don't have to do any of the mathematical work. In fact, the system does it for you. So you just subject it to a system and you get a graph as well. Um, and previously, even for GCMS as well as for LCMS, you do get graphs which will, I would say, properly produce results of each and every analyte, that is, every component which needs to be analyzed in the form of a graph. So when you calculate the peak height, when you get a specific peak and you calculate the peak height as well as the peak area, you will be able to quantify how much of that drug is present as well. So MS, I would be explaining the advantages of MS at a later stage. But as far as UV visible spectroscopy is concerned, it's one of the most simplest techniques which is used by a scientist, not just in the forensic field, but even at the pharmaceutical field as well. And I would even say, even most microbiology sectors also have started using UV visible spectroscopy as an effective technique. So that's for this paper. Now let's take a look at the advancements. Now, I've mentioned about drugs being in powder form, in pill form, and so on. But there are studies which have been done where drugs have been retrieved from water. And now you might be thinking, how is that so? Like, who's going to mix drug with water just like that in a natural water source? Now, there was actually a very I wouldn't say it's a very old case, it's quite recent. There was a case where a person was on the run from law enforcement agencies. And so, to hide the evidence of the powder, the person actually went to a water body close by, I think it was a river, and dumped the entire components, thinking that this might dissolve. And yes, it did dissolve. So, the person thought, ha, today is my lucky day. I'm not going to get caught by the police. But that's where you guys come in. That's where the forensic experts come in. They said, no, you are definitely getting caught. They basically took the samples of water and subjected it to HRMS, which is high resolution mass spectrometry, and voila, the drugs were actually detected. So in this case, HRMS proved to be a very effective technique. It is in fact so specific that it can detect even 0.001 atomic units of that specific weight of the chemical drug. So in that case, it can also detect multiple analytes as well. So in this case, if the water sample is going to contain multiple classes of drugs, this is the right technique that you should go for. So this was one of the papers which was done, where illicit drugs is nothing but drugs which are not supposed to be ingested in the legal field. So this was also done. So if you guys are interested in knowing how the instrumentation works, I will quickly summarize. Basically, you have the sample which is injected in this case and it gets vaporized. On vaporization, it produces certain ions which get accelerated towards a magnet. And that magnet is essentially going to deflect the lightest ions first and to a greater extent. And the ones that are lighter will get detected. And MS is a coupled detector, that is two means of MS are present, whereas MS is simply a detector which is used to detect components and separate them on the basis of their mass to charge ratio. So M by Z ratio is what we call it. So on the basis of the mass to charge ratio of a specific drug, this is how it can be quantified and detected in the first place. Um, Last but not the least, you have a technique which is known as miniature mass spectrometer, uh, or which is using ambient ionization. So here, cannabinoids, that is components of cannabis sativa, which a lot of you must have heard. This was also detected. This was using a very rapid technique as well. So the advantages of this technique is it's rapid. Okay, it has high means of analysis, and unlike GCMS, in this case, you do not need to treat your sample with any chemical as such. In fact, samples like urine, blood, etc., just as going through a simple extraction procedure, the extract alone can be subject to the system. So I would say this has been a great advancement in the field of forensic science, especially in the field of forensic drug analysis. And I would say that I have a lot of hope in terms of analysis being a little more specific, a little more robust and rapid in the future. And I strongly believe that the students who are engaging in research today will definitely bring results for improving the analytical techniques that we might require in the future. Because as you know, crime never gets simpler with time. It just gets more complex with time. So that was my webinar for today. Thank you so much. I'm open to questions.
First of all, thank you very much, ma'am, for your such a interesting and useful information. So we have some questions, ma'am.
Sure, sir.
Yes. So what analytical techniques are commonly used in forensic drug analysis?
So, just to have explained, basically a lot of techniques that you go for are usually U visible spectroscopy. You do go for LCMS, which is liquid chromatography, and you also go for GCMS, which is gas chromatography. At a simpler level, you can also go for thin layer chromatography because there have been studies which are done where, just as I mentioned, cannabinoids being detected. Cannabinoids have also been detected by using TLC as well. So TLC is nothing but thin layer chromatography where you just have to plot certain samples on a silica plate and then keep it to develop using a mobile phase. So I would say it will get more complex as it goes, but these are some of the common techniques which have been used.
Okay, ma'am. The next question is, what procedures are followed to ensure the integrity of evidence during the drug analysis?
So one thing during drug analysis is never assume that this could be the drug that you're looking for because all of us are very quick to become too positive about a specific case. Essentially, we want to say that, oh, this is definitely cocaine. The authorities are going to be very pleased with me that I saw white powder and it's cocaine. But that's not the case. So I would say when you go into a crime scene, you will go with a very broad state of mind. You will look at every evidence as if that's crucial to the crime and to analysis as well. As far as laboratory analysis is concerned, I would say you will take certain precautions. The kind of evidences which you might get from a crime scene might not come in larger quantities. Um, it could come in traces, it could come from biological samples, and essentially, ethically, also, you wouldn't be asking the patient for more than a few milliliters of blood. So, in that case, using a small concentration of evidence, you should be bringing about huge results. So, if you are going for analysis, you should usually start with a non destructive technique where you can easily retrieve the sample and then subject it to further analysis, and then you can go for destructive techniques. So I would say that's the case.
Okay. So there are some questions from YouTube also.
Yeah,
What
sure.
challenges do forensic scientists face is when analyzing drug evidences obtained from the clandestine laboratories?
So clandestine laboratories, for those of the students who are not aware of the term, they are usually laboratories which are underground, which are usually hidden. It's managed by huge private party of people, a huge party of criminals, I would say, where they would essentially manufacture these drugs illegally. So they are usually concentrated in certain areas as well. That's a clandestine lab. And most of the sting operations that you must have seen in movies, you must have also seen in YouTube, on YouTube as well. They basically talk about investigating a specific crime scene and maybe busting the crime ring by entering the crime scene, and you will see a lot of machinery present for producing that kind of drug, etc. So some of the challenges I would say is, in terms of communication between culprits, they're very quick to hide evidences. So a clandestine lab can be small and it can be huge as well. A clandestine lab can also be created within a house, in someone's kitchen as well. So essentially, when the person comes to the crime scene, if they have received news of law enforcement entering that specific area, they are quick to hide. And the second thing about clandestine laboratories is its efficacy in destruction. So a lot of people, again, in terms of hiding the clandestine lab, they essentially would explode the crime scene as well. So I would say that's a lot of work for the forensic expert. Now they have two crime scenes to look at. They have a case of burning the crime scene as well as the crime scene itself. These are a few challenges I would say, but apart from that, a clandestine lab is actually a very good source of getting a large quantity of the evidence. So I wouldn't say there are huger challenges, but this is some of the these are some of the challenges I would say.
Okay, thank you. So, how does forensic drug analysis contribute to understanding drug-related crimes and trends in society?
Um, so purely in terms of forensics, you have case of homicide. The thing is, people know that drugs are the quickest means for getting rid of someone that they hate, unfortunately. And in such cases, you can see in case of homicide, trying to get rid of a person by causing death, they do administer such drugs. So in case of accidents, accidental poisoning, or I would say homicide, suicides, even committing something which is widely popular, unfortunately known as date rape, as the word suggests. Date rape is nothing but administering a drug to an individual, usually in clubs and so on, where they'll add the drug into the person's drink. And usually, what happens in such cases is it might change the physical appearance of the drug to an extent, but if the person is not cautious about the drink that they are having, what happens in such cases is they consume the drink, and the next morning they unfortunately wake up remembering either having flashbacks of being sexually abused by that culprit, or they don't remember anything at all, but they have severe wounds on their body. So in such cases that is a challenge because the whole meaning of consent steps in. So the law enforcement would ask, was it consensual, was it not? But that's where forensic chemical analysis steps in. um, Obviously the person who is aware of consuming such drugs would be aware of the quantity that they have taken in. But for a person who has not, a person who is completely unaware of the surrounding in which they are in, suppose if they are not in a club but they've been subjected to or they've been administered with a club drug, then that, after verifying facts, you can understand whether this person themselves have consumed that specific drug or whether it was actually administered in their drink or through their food. So in such cases as well, you can definitely conduct drug analysis and quantify as well and see, if this is the cause of crime, if there are multiple culprits which are present, or if the person themselves are lying or not. So I would say this is the answer.
Okay. So, one more questions. Drug analysis done in hair, how much accurate it will be?
Pretty accurate. If you use a very good instrument, you're bound to get very good results as well. And I would say if you talk to most seniors in your university, they do go for hair samples for their dissertation because they do know that biological samples like hair, especially hair alone plays a very good role as a biological specimen. But in terms of, I would say, forensic chemistry as well. Like I've mentioned before, most of the drugs get concentrated along the roots of the hair. So in such cases, if you do perform a quick, robust, rapid analysis of drugs using hair samples, I am sure that you will be getting satisfying results. So yeah.
Yeah. I hope all participants get their solve their query. So thank you very much, ma'am. Your topic is really
Thank you
very
so much.
interesting, and we learn so many things from you. Again, thank you very much, ma'am.
Thank you so much, sir, for this opportunity.
Thank you, ma'am. So, dear participants, our next webinar is on 30th March 2024. The topic is AI-based forgeries. So, registration link and Zoom link will be shared on your registered email ID. Thank you very much, all.